And we are starting with an interesting talk of Anna Ronikier from Polish Academy of Sciences, who will tell us about importance of type collection in taxonomical studies on an example of Cyphoptychium genus.
Thank you.
Cyphoptychium polyschismium.
It's not exactly only polyschismium, it's only example picture. But okay, so since I'm a traditional taxonomist, I would like to put your attention on the importance of including type specimens in phylogenetic studies. Maybe for some of you, all of this I will be talking about may be obvious, but based on the survey of published papers and also my own experience, I feel it is very important to all of us to put more attention on how to treat type specimens and original materials. And to illustrate this issue, I will show you some of results obtained by our team, that it's me and Paulina Janik, in collaboration with other colleagues. And by the way, Carlos Lado, who is mentioned here, the first as our collaborator, he couldn't attend this Congress, but he asked me to pass his greetings to all of you. So we had the pleasure to work with several people during the last several years, so I will show you some of those results we obtained. But before I will go to the detailed examples, I would like to start from the beginning, so to define the type specimen. So there are several codes of nomenclature. One of these is nomenclature code of International Code of Nomenclature for Algae, Fungi, and Plants. And in this code, in the point 8 of the preamble, slime molds are mentioned. So we can be sure that myxomycetes fall under the provision of this code. And in the glossary of the code, you can find the definition of the type specimen, which is further explained in the article 7.2. So the nomenclatural type is the element to which the name of the species of the taxon is permanently attached. In other words, the type specimen is usually a single specimen based on which a given species has been described. And if you are more interested in problems of nomenclature of myxomycetes, you can go to our paper published a few years ago together with my colleague Adam Halamski. And in this paper, we explain why myxomycetes shouldn't go under the provision of the zoological code. And following the code of nomenclature for algae, fungi, and plants, you can find more. This is the best, of course, the best publication where you can find information on type specimens and nomenclature of myxomycetes in general. So for instance, you can learn that living culture cannot be designated as a type for a myxomycetes species. What can be done for animals, but they fall under the provision of the zoological code. So some protistologists suggest that myxomycetes should go under zoological code, but it is not the case. So the first question when we want to study type specimens is how to find them. And for the newly described species, it is not so difficult because the current rules of the code requires a direct link to the holotype and to the specimen. So in the case of this Physarum andimum, which we described together with Carlos Lado a few years ago, you can see that the holotype is deposited in Emma fungi with a number of herbarium collection, and the isotype, which is a part of the holotype, is deposited at Cram, which is our herbarium in Krakow. By the way, now Physarum andimum has been transferred to the genus Craterium, so by Carlos Lado team, so now it is called Craterium andinum. But the isotype of the species you can find in our herbarium in Krakow, and if you find the same number that is mentioned in the protologue, which is 1553, you can be sure that you have the isotype in your hands. Since our part of our collection in Krakow is digitalized, you can find this picture in the internet also, and you can find also the specimen inside, which is available at the platform rcin.org.pl. And when the species was described a long time ago, it is not that easy. So as an example, the Anemma harvey, described from North America in 1891, in the protologue, you can find only information on the locality and name of the collector. In the BPI, the largest herbarium of the United States, you can find 3 boxes with the information fitting the protologue. So which one is the type specimen then? They are all syntypes. So syntypes are any part of original material that can be designated, from which electrotype can be designated, and electrotype can be designated from original material when there's no holotype. And I would say that from these 3 collections, the first one is probably the best candidate for electrotype, since it lacks question marks, and also the annotation types lacks question mark. So when we have a look at this collection and we look inside, there's a nice specimen. And when we examine it microscopically, we can see the penicillate capillitume with spirals, what means that it represents Prototrichia metallica. So the conclusion is that the Anemma harvey is simply a synonym of Prototrichia metallica, since Prototrichia metallica was described 2 years earlier, and its name has priority. But in another herbarium of the Academy of Natural Science of Philadelphia, there is another specimen, which also information on label fits the protologue. And also the collection date of this specimen was earlier than the description of the species. So it's also another candidate for electrotype. And when you see inside, there's also a box with a nice collection, also annotated with type. And additionally, it is annotated with handwriting. And with the great help of the curator of PH, we could identify this handwriting as belonging to George Rex, who is the author of the species. In this case, we could say that probably this species was in hands of George Rex when he was describing it. And when we look to the microscopy of the species, it has smooth capillitume, is a little bit different. So the Anemma harvey is simply the Anemma harvey. So we must be very careful when we study original materials, because you can never be sure that you have all pieces of original material in hands. And also, when you decide to designate electrotype, you can change the meaning of the species, like in the case of the first collection I showed you, because the author who first designates electrotype must be followed. And also, if you don't want to designate electrotype, but you sample for phylogenetic studies from the Rome collection, you can also change the meaning of the species or even genes. Another problem when we met, when we want to sample from type specimens for phylogenetic studies, is the sampling for DNA analysis. The problem is that the most common isolation kits used in myxomycetes requires removal of several sporocarps. So in the case of the holotypes of types, which are usually scarce, when you remove all specimens from this collection, nothing remains. And it is, of course, forbidden because herbarium rules forbid to destructive sampling from type specimens. So simply, type specimens are unavailable for such sampling. To overcome this problem, our team tested several protocols. One of them was DNA as a plant mini kit, which is the most commonly used. And we sampled from several sporocarps. Then we used the same isolation kit and we sampled from one sporocarp. Then we used a investigator kit, which is devoted to sample from small amount of material. And it was advised us by our colleagues, mycologists. And these 3 procedures are based on the genomic DNA isolation step, and then after subsequent PCR, you can get the target DNA marker. But we also invented a totally new protocol for which we need only a portion of spores. So we simply pinch the sporocarp with an injection needle, and those spores which attach to the needle are taken. And then we, with the omission of the genomic isolation step, we directly obtain the given DNA fragment with Fireplant direct PCR master mix. If you are more, we had this fourth procedure turned out to be the most effective. So we studied it on several species from both bright spot and dark spot species, and we got 95% of success using this method. If you are more interested, all the results were published a few years ago in PJ. And using, in fact, this fourth procedure, which turned out to be so effective, is also very quickly, very cheap, very convenient. So we now use it routinely in our laboratory for barcoding of specimens, which is really very fast. And this protocol, the use of this protocol opened the new doors of sampling from type specimens, since it is practically no destructive. So we take only a few spores, which is less destructive than making a microscopic slide. And with this protocol, we were able to obtain barcode markers of dozens of type specimens. The oldest of them is Trichia sordida, which was 43 years old. I mean, the collected 43 years old before sampling. So simply using this method, we are able to obtain barcode marker from species described from during last 40 years. So it is, I think, pretty nice. And now I'm going to show you some of our results. And together with Carlos Lado and Ivan Garcia Conjillos, we studied niviculate myxomycetes from the Andes. And apart from the description of two new species, we also confirmed that Perigena megaspora is an independent species. Perigena megaspora was described based exclusively on morphological characters a few years before. By the way, now its name is Guglielmina megaspora, which was changed also by Ivan Garcia Conjillos from the team of Carlos Lado. And here you can see the two-gene phylogeny. And here is the paratype of Perigena megaspora, which closed the sister lineage to the newly described in this paper, Perigena patagonica and Perigena vermicularis. So we confirmed it to be independent species. In another study carried out together with Miriam Dehaan and Andreas Kund, we focus on the genus Lepidoderma. And here we confirm that Lepidoderma stipitatum represents Diderma floriforme. It was previously suggested by Michel Poulain, Marianne Meyer, and Jean Bozonnet. And we could confirm it based on the isotype material. And here is you can see the sequence of the isotype. Here is another collection of Lepidoderma stipitatum, which was morphologically identical to the isotype. It was sent us by Henrik Goetsche, and also the sequence of Diderma floriforme. And you can see that they are really conspecific. And also when you look at the morphology of these two species, you can see that the only difference is the structure of the peridium, which is crystalline in Lepidoderma stipitatum and Diderma-like in Diderma floriforme. And when you compare the spores of these two species, which are unique among myxomycetes, because no other myxomycetes have such a spore, you have no doubt that they are the same, even solely on morphology. The second species we verified was Lepidoderma crassipes, which we confirmed to represent Lepidoderma tigrinum. And again, in this phylogeny, you can see the sequence of the isotype of Lepidoderma crassipes. And the second specimen we got from René Skrzypczak from France. It was also identical morphologically to the isotype. And all other sequences you see here in this pink clade are all known sequences of Lepidoderma tigrinum generated by us or taken from gene bank. So you can see that they are really exactly the same. And also based on morphology, again, the only difference between these two species is the structure of peridium. In the case of Lepidoderma crassipes, its peridium is covered by sand-like, very small, tiny scales, and Lepidoderma tigrinum, yes, you are familiar with this species, I guess, has star-like plates. And in this paper, we also studied other species, other type specimens of two other species, Lepidoderma neoperforatum and Lepidoderma perforatum. And we also confirmed they are independent, what was questioned a few years before. And also we transferred all species belonging to Lepidoderma apart from the type species, which is Lepidoderma tigrinum, and then was transferred to the genus Diderma by Martin Schnitter's group. So we transferred all other Lepidodermas to the Polyschismum, the old genus. And another study we are doing together with Miriam Dehaan and started several years ago, we would like to build a reliable type-based phylogeny of the genus Lamproderma. And here you can see the first results. This is the phylogeny based on the barcode marker. And here we used only sequences of Lamproderma diahaopsis taken from gene bank, all unique sequences, also all generated by us, and sequences of type specimens of Lamproderma and diahaopsis. And I will show you now some examples. The first diahaopsis, which was already shown by Martin and Angela, that it was also suggested by Anna Maria Fioredonno before that it belongs to Lamproderma. So we also wanted to verify this. And here is one place when diahaopsis occurs. Yeah, you can see this. I will show you these places in the tree. And this is diahaopsis reticulospora with its type sequence here. Here is diahaopsis pauxilla with the type sequence here. And it is close to Lamproderma zonatum with the type, holotype of Lamproderma zonatum here. And another place in the tree, diahaopsis kowalski with this type specimen here, which is, by the way, close to Lamproderma pseudomaculatum, which holotypes is here. So you can see that diahaopsis is dispersed, that you have the same results. So we also confirm, based on type specimens, that diahaopsis belongs to Lamproderma, of course, in the present meaning of this genus. And also, here I would like to emphasize that we, again, must be very careful, because diahaopsis is, as you know, Lamproderma without columella and stalk. And some Lamprodermas can form such malformed specimens. So I was very surprised to see the specimen of Lamproderma acanthosporum without columella and stalk. So when you have the specimen which looks like diahaopsis without comparing it to the type sequences, you can never be sure if you have maldeveloped Lamproderma or real diahaopsis. And with some other examples in this phylogeny, we, for instance, confirmed Lamproderma argenteobrunneum to be an independent species with its isotype sequenced here, and Lamproderma cacographicum with its holotype. They are sister species in our phylogeny, which is not surprising based on morphology. We also confirmed Lamproderma lycopodiicola with two sequences of two paratypes. And here, again, we must be very careful when we have paratypes only, because once I loaned 2 paratypes of Comatricha, and one of them was Meriderma. So remember that the most important is holotype, isotype, because paratypes are only additional specimens, and also mistakes are possible. And then we can see also Lamproderma vietnamense, which forms a separate lineage. And with Lamproderma zonapulchellum, we cannot say so, but not because it is not well described, not independent species, but simply because we don't have a sequence of the type of Pulchellum. So we cannot confirm it with this phylogeny. So with the type specimens, we could do it. And then the most spectacular, I think, example of how important is to study type collection is another species, which is Lamproderma eneum, which appears here in the very nice monophyletic clade. Here you can see a lot of sequences of Lamproderma eneum from gene bank, which is close to Lamproderma andinum, which is type specimens here. But there is another place in our phylogeny when we have Lamproderma eneum, again, monophyletic clade of Lamproderma eneum. And where is the real one? The real one is here, because here is the sequence of the holotype. So with the holotype, we can really calibrate, let's say, the phylogeny, because it defines the place when the real specimen is. So in summary, what is the most important in our phylogenetic studies? First, to follow International Code of Nomenclature of Algae, Fungi, and Plants, and follow all its rules. Then to be very careful in designation of lector neotypes and in the interpretation of original materials. It is very tricky, and also some collections are transferred from one herbarium to another, so you'll never be sure where you can find the original material. And use non-destructive sampling of DNA from type specimens, and also, whenever possible, use sequences of type specimens in phylogenetic analysis. And just for the very end, I would like to show you our idea, the idea of our team to create the type specimen database that we would like to place in the new webpage of our team, which is under construction now. And here's the example of one of the species. We would like to link it to the Nomen Myxomycetozoa database, and also with the sequence of the type specimens taken directly from the gene bank. The link will be directly for the sequence of the holotype, because sometimes, as you can probably see in gene bank, you can have the name Lamproderma species, and you don't know what is it, and it can be the holotype specimen of a described species. And also, we would like to illustrate this type specimen with macro morphology, with microscopical characters, and to give the link to the publication in which the given specimen was examined. So we have first pages of this planet type specimen database, and for the beginning, we would like to present here our results, but we would like it to be in the future a place open for everybody who would like to put the results of their own study here. So thank you very much.
Any questions, please?
Thank you for your talk. I will read a question from our online participant, Ivan Garcia Conchilos. He says, such a great presentation. Thank you. I would like to know what is your opinion on how to proceed when we cannot obtain DNA sequences from type specimens, most usually because of their age.
Yeah, so there is a possibility to find another collection determined by the author, but we must be careful if the author didn't make a mistake, and to sample from the other original material, which is not holotype. But when it is old, to degraded, to obtain DNA, we cannot do anything. In fact, we have to wait until we have new tools to obtain sequence from degraded DNA. But of course, for the time being, we can also choose the specimen which really fits the diagnosis. What sometimes is also difficult, especially for old species where the diagnoses are very short and can fit several species. So this is a real problem. Yeah.
Now, from myself, I know that it is possible to override an electrotype if some better material, proper material was found. And do you know of any such cases in myxomycetes?
No, it is not allowed if the better material is found. Once a electrotype is designated, it must be followed. There are some occasions where it can be changed, but only if you prove the electrotype doesn't fit the protolog.
In such cases.
But it's not that you have something what is nicer and nicer fits the protolog, but if you found that electrotype belongs to another species.
To have the same questions a little bit more precisely. Let's assume you have 100-year-old, not morphologically nicely characterized and nicely preserved type specimen. The only problem is the DNA is highly fragmented. You can't do Sanger sequencing. Could you try to find authentic material which is fresh and new and design a lectotype or not?
No, but lectotype can be designated only if there is no holotype.
Okay, that's my understanding as well, which means a morphologically well-preserved holotype, in other words, would block further research, and that's what I don't like and want to ask for a way out, since you can only relate to the holotype based on morphological characters, and that's all right now.
Yeah, I mean, theoretically, yes, but as you know, and as you can see in published papers, research is not blocked because there's no holotypes in most phylogenies of fungi, plants, and so on. So we still, I mean, this is the very strict attitude, that to be really very clear, very sure, I mean, the elegant, you should always, whenever possible, include type specimens so that you are completely sure, because the type specimens is the reference material for the species. But of course, it depends in practice. It depends on the situation, because, for instance, when we have Lamproderma acanthosporum, which is so easy to identify that anybody can do it, taking even one spore, then you probably don't need to have holotype as much as in the case of Lamproderma arcerioides or splendens or Aneum. In this case, for instance, in Aneum, you cannot say anything about Lamproderma Aneum when you have no reference material, because you have two separate clades, and one of them is Aneum, but which one? So in practice, for complex species, type specimen study is particularly important. And also, if you have very old material and you cannot sample for DNA, you can also base on morphological characters, and you can find the morphologically identical specimen and treat it as the reference material. I mean, there is no rules for that, but only just for your studies. But this is your subjective experience. So if you really can recognize another specimen as belonging to this species, you know, it is subjective. You can do it, and somebody else is not able to notice such slight differences because it depends of our ability to recognize features. Yeah, so it is more subjective. So of course, in practice, we have to somehow ignore type specimens in regular studies because we could stop. I mean, our research would stop for, I don't know, hundreds of years searching for type specimens. Yeah. So yeah, we just would like to be the most close to the type specimens. Yeah.
Could it be an idea in the case which Martin described to design the epitype from the newer collection, which would be sequenced, if it is really morphologically close to the well-preserved but too old holotype?
You mean when the holotype is present?
The holotype is present. Is it allowed to design additionally the epitype from the newer collections?
Yeah, it is allowed to, for the material which is not informative, sufficiently informative, you can designate no type. But the recommendation of the code is that when you have specimen and you can identify it with this name, you shouldn't designate no type because you still have specimen which you can define. It can be done if the lectotype is the picture, what sometimes is the case. So because lectotype can be a specimen and also can be a illustration. So if you have lectotype and illustration, it's obvious that you cannot sample from it. So then you can designate a neotype. Yeah.
Okay.
I'm very happy to know that more people are studying molecular aspects of myxomycetes, and what the research, the research group has done a great job actually. My question is, what gene that you use for the sequencing? For the phylogenetic genes.
Yeah, we usually use SSU rRNA gene, which is considered to be barcode marker. The parts of this gene, which is considered to be barcode marker. And since it is not very long, it is pretty short, it is easy to, relatively easy to obtain from old specimens. So that's why we rely on this marker. We also tried to sample another genes from type specimens, but it is very difficult. We couldn't obtain nice results. So the problem is also that these type specimens, sequences of type specimens, cannot be included in multigene phylogenies because we have only one gene. Usually one gene obtained from them. Of course, if the species are fresh and newly described, we can have more genes. Yes. But for older material, other genes which are present in less repetitive copies are more difficult to obtain. Yeah?
So I have a small follow-up question, which is how can you make sure if that is because of the gene, the mark isn't not specific enough, or it is because of the effects of the specimen types? Each specimen itself already has some kind of diverse in the gene sequence, right? Naturally. So how can you tell if it's because of the non-specific, I couldn't answer that, but because of the specificity of the gene, or because of the actual effects of the specimen types?
First, this gene was before proposed by, based on numerous research to be a good barcode marker because it based on phylogeny based on this gene give us reliable phylogeny. So we can really, for in most cases, yeah, in general. So it was tested that it is a good barcode marker. And also, of course, it is variable. So if you, for instance, go to this clade of Lamproderma anneum, yeah, there are all of this we consider to be Lamproderma anneum, yeah, all of this. So we can see that it is diverse. Yeah? So it's not only one sequence which has to be identical, but it has some changes, but they form monophyletic clade, which is the most important because monophily is the most important in interpretation of separate species. So they might be monophyletic. Yeah.
Any more questions?
Yeah, I'm just a little bit quick. I just wondered, how do you think about the boundaries of the gene? The genus. What is the Lamproderma? This means that the Lamproderma columbinum is a type of the genus, but it's very old and very small. Short description on it, and there's no material, I think. So if you think about what is the Lamproderma, as Andrea also said, yeah, how do you think about it? If there is no specimen holotype or anything, how do you think?
Yes, I think we still need more data and more wide sampling, and to put it in the larger phylogeny, including Physaurales, to see, because Lamproderma is probably polyphyletic, and it probably will be divided in several genera in future. But to do this, we need to have really reliable results, which we don't have now, I think. At least I would say so.
Okay. Any more questions, remarks?
Just a short comment. I really like the point with the type specimen database. It's what the miss is a link between known mix, which is pretty much known collaterals and so on, and the gene bank, where you indeed often have not the proper identifications for your sequences, since it's really hard afterwards to change a denomination, and you can also not put int names, which means preliminary names, in gene bank.
Yeah.
So to establish the link later and say, okay, this gene bank reference belongs to that holotype would be a really, really useful thing.
Yeah. And I would also like to stress that we should try to put the sequences of holotype with the name of the holotype. I mean, the name of the species. So not preliminary names, so that everybody can take who can take sequences from GenBank immediately see that it is at least this species. Because in any cases, you can find a holotype of one species, and in GenBank, it is under another species or under Lamproderma or Diderma, whatever SP. So in this case, you can, it is not so easy. Of course, you can find this information, but it is not this straightforward.
Where you put like all this information, the fields. Before you upload the field month, you can field.
Yeah, you can. Yeah, this is. Yeah, the person who produced the sequence and submitted to GenBank, he or she fills or needed information. And then it depends on you if you put, for instance, Lamproderma andinum or Lamproderma sp, and then everybody can see what you entered, yeah? I don't know if it's place for two names there. But if it is the holotype,
are submitted the sequences of species that is not described, but yeah, you are preparing publication and submit sequences in parallel to it for the description of new species. Even if you provide the name of the new species with the sequences, GenBank then changes the new epithet that is not yet published to your initials and the year. And then it's up to you when the publication is already accepted or published to go back to GenBank and
change the name.
That's a holotype. The holotype is already the name, but then you can use.
No, but the holo. Yeah. So in the case you find that already existing in the GenBank sequence belongs to the holotype of your new species, yeah, then you cannot, I mean, you probably cannot change this name, yeah, if I understand. But when you enter this new sequence to GenBank, it's you who name the sequence, the specimen. But also when you change the, for instance, the taxonomy of the species, I mean, you found the two species conspecific, for instance, so we cannot change the name of the holotype because holotype is permanently attached to the species on which was described, yeah? So although we consider, like for instance, in the case of Lepidoderma stipitatum, the holotype remains Lepidoderma stipitatum, but we call the species Lepidoderma diderma floriforme, yeah? Like that.